Welcome back to Plant Science. This is Chapter 3. We're going to talk about plant structure and some organizations of the cells within the plant. We're going to essentially form a plant glossary. So let's begin talking about cells. We know that cells are the basic building blocks of living things. It's called the cellular theory of biology. As I've said before, a theory is as good as a law in physics. It's been tested a lot of ways, and we have decided that this is, in fact, the way that the world works here. Now, cells were discovered by Robert Hooke in 1665, so they've been around a long time. We've known about them a long time. He built a compound microscope, ground some glass and polished it, and looked under some cork, which is the bark of an oak that grows in Spain, very large cells. He's able to see that it looked like little spaces where monks slept in a monastery, and they were called cells, and so he called them cells. But we also know that there's a great variation in size and function in cells, and we will take a quick tour through some of these. They're not certainly all the same in our bodies, and they're not the same in plant bodies either. Now, we can begin by splitting the cells up into two groups, the prokaryotes and the eukaryotes. The prokaryote, pro means before, and this is before there was a nucleus, right? The DNA just floats around freely, and these are considered to be ancient and primitive organisms, usually single-cell organisms, no subcellular compartments, or they're at least considered to be very primitive. Bacteria fall into this category, but everything from yeast to corn plants to people are eukaryotes. EU means true, and so these have a true envelope around the nucleus and the subcellular components and compartments, and this is what we're going to be talking about mostly in higher plants, although we will talk about prokaryotes just a bit if we ever talk about nitrogen fixation. That is performed by a prokaryotic organism. Well, what's in the cell? Well, let's take a quick spin through what's in a cell. What are these organelles? An organelle is a small organ contained within the cell. First of all, the guts of the cell is held in by the outside membrane called the plasma membrane or the cellular membrane. It decides what comes in and what goes out of the cell. And then there's the nucleus, which is the part that contains the DNA and is the brain of the cell. Plastids are bags, if you will, or sacs within a cell that hold different things. Leucoplasts hold volatiles that are used in flavoring or in pharmaceuticals. So if you smell a nice flavor in a flavoring, a food flavoring, it might be coming from the sac inside of a cell. Idioplasts are for embryonic plastids, and also idioplasts appear. I'll tell you what those are is you see those if you've ever parked a car on a grass or laid a board on grass for most of a week, and then you pull it off and you'll see that things are yellow under there. Those are chloroplasts, which is another type of plastid. A chloroplast which has not developed because those need sunlight to develop, and so they are embryonic plastids. And then the chromo means color, so chromoplast is where some of the colors are kept in the plant cells. Chloroplast is the green plastid. It gets its own category, and it's where things happen. It's where photosynthesis happens, the magic of plants. Mitochondria are the power plants of the cell. We have mitochondria. That's how we are able to move and use our brains, is because that's where we extract energy from energy-rich molecules, and that's where plants do the same. A vacuole is sometimes the very largest thing in a cell, and it is sometimes just a place to store things. It's like a plastid. We're not going to go over too much endoplasmic reticulum or Golgi apparatus, but those are also parts of the cell. And then the cell wall, sometimes only the primary, sometimes primary and secondary, and you'll see that's very important for agriculture purposes. And then the cytoplasm is everything in between inside the plasma membrane. So it includes all these organelles we've been talking about. The nucleus, the plastids, the Golgi apparatus, and everything else is in the cytoplasm. Cyto means cell, and plasm means fluid. So literally, it's the fluid of the cell. A quick visual, very basic visual. You see that at the top is a nucleus where the DNA is contained. You see the red line just inside the dark, thick black line. That's the membrane. That allows passage into and out of the cell. It's semi-permeable membrane. The dark black line is the cell wall. The mitochondria there is the power plant. The plasmodesmata, which we did not mention on the previous page, are little channels that connect cell to cell. We think they're used for communication. The green ovals are the chloroplasts, a plastid where photosynthesis happens. And then the big vacuole, often it's the largest component of a cell. In the middle, just a big sac where the plant can keep things. Well, let's compare plant and animal cells. This will be a pretty important part of the test and understanding of plant science from animal science. The differences between plants and animals is that plants have a cell wall. Okay, we don't have a cell wall. We're animals. If we had a cell wall, we couldn't move. Cell walls are what keeps plants upright and stiff but non-movable. We don't have vacuoles. They're very large in plants. If they are in animals, they're very small. And then certainly plants only have chloroplasts where photosynthesis occurs. That does not occur in animals. But what are the similarities? Well, we all have nuclei, cell membranes, mitochondria, endoplasmic reticulum, cytoplasm, so much more in common than not in common. So you should know those as well, but you really need to know those differences: cell wall, vacuole, and chloroplasts. Well, we can divide the plant up into what types of cells that we're looking at. There are two types of tissues. One is meristemic or meristematic, and one is non-meristemic or non-meristematic. The meristems are the growing points. And if you imagine a plant, there's a little diagram here on the right. If you imagine a plant growing, in this case it's a little oak tree, you see that the very tip of the branch is the apical meristem. That's where the growing point is, but that's not the only growing point. There's an axillary bud in the axil between the leaf there and the stem. And in the axil of almost every leaf and stem intersection on the upper side is a dormant bud. And in case the top of that plant gets knocked out or cut out, then that dormant bud will wake up and start to grow and become the new top of that plant or that tree or whatever it is that we're talking about. Okay, so there's two growing points right there. One is active, apical, and one is dormant, axillary, that could become active. The intercalary meristem is only in grasses, and it is an unusual meristem, but you'll understand the way it works when I explain it to you. It's not at the tip of grasses, it's at the base of grasses, oftentimes just slightly below ground in what we would call the crown of the plant. Imagine that you mow the grass, and every time you mow the grass, you're cutting off leaves, but you're not cutting off the apical growing point or even the axillary growing points. Those are at ground level or slightly below. It's like getting a haircut. Okay, you cut your hair keeps growing because the growing point of your hair is in your scalp. And that's the way grasses grow. The growing point, the meristems are there, they're just down in that crown. So if you overgraze a pasture for too long or with too many animals, or you scalp your lawn with the mower too low, the setting too low, you will damage that intercalary meristem and cause the grass plant to be harmed, or even die. Okay, so grasses are not like other plants. And then the fourth kind is a lateral meristem. That's the vascular cambium, or some people call it the cork cambium. Those are on woody plants like trees. If you imagine, a tree gets taller every year, but it also gets fatter every year. And the fatter part of the growth of the tree is what we call the lateral meristem. That's that bright green cambium layer just beneath the bark. And that's a ring that grows quickly in the spring and summer, and then not very quickly in the late summer and fall. And so those are the rings that we count when we're counting the age of a tree. We're counting the growth habits of that lateral or secondary growth meristem, the cambium layer in trees. This is an example of a little plant, a little garden plant, coleus, and this is a transverse section through the coleus. Imagine if you were to take a carrot and split it down the long ways. That's a transverse section. In this case, we're looking at a transverse section of a tiny little section at the top of a herbaceous plant. This plant does not lay down wood. It's a coleus. The dome at the very top, run my mouse over there, the dome at the very top is the growing point. That's the apical growing point. Those cells are rapidly dividing, and then once they get down below that area, they start to blow up like balloons. So the upper part is like making the balloons, and the lower part is like expanding the balloons. And then you can see that the two seeming arms or ears or horns that stick up above it are really the very young leaves being formed that will expand in a few days or weeks to become full-fledged leaves that you can see with your naked eye. But right now they're just a few cells in size. You can see the dark bands showing up in these primordial leaves, and those are the veins, that big vein that runs down the middle of the leaf. It's there. Then if you look a little lower to the right and to the left, you see larger leaves, parts anyway of larger leaves, and then in the base of the leaf and stem axil is a dormant lateral bud on either side. And so we can see two growing points, types of growing points right there, 3 growing points in total. Well, that was meristematic or growing tissue. And then the other type that we can have are permanent tissues. And these are no longer dividing. They are static. They have become what they're going to become for their lives. We can divide these types of permanent tissues up into 3 categories, or just a few more. But anyway, simple, which are uniform, one cell type. They're pretty much the same. The smallest, the thinnest cell walls, the weakest of the 3 types I'm going to tell you, are the parenchyma. You say that parenchyma, like for instance the epidermis on the upper surface or lower surface of a leaf. These are thin walls. They're undifferentiated. They're not specialized. They are somewhat succulent, and those are thin-walled. And then there's collenchyma. The collenchyma is a thicker primary cell wall than the parenchyma. It's a little stronger. The plant has to make a little more investment in these leaves. The edges of the leaves have to be tougher than the middle of the leaf, right? So the leaf margins. The petioles, the part of the plant that looks like a little stem that holds the bottom of the leaf where it attaches to the stem of the main plant, has to be mechanically strong and reinforced. And then some things like fruit rinds, cantaloupe rinds, orange rinds, and then the veins of plants. These have to be a little bit thicker, a little bit stronger mechanically than simple parenchyma cells, which are the spongy things inside of leaves, for instance. And then finally, we've got the sclerenchyma. The sclerenchyma is the very hardest of the 3 types of permanent plant tissues. They're the thickest cell wall of the 3 types. These actually have a secondary cell wall as well as a primary cell wall. They are strong, and they usually have a mechanical role beyond that of veins. They're elastic and resilient, and these comprise the economic and harvestable components of crops such as hemp or flax or even sugarcane. Hemp or flax are grown for fiber, so it makes sense that they would have strong sclerenchyma cells. And sugarcane is a really large grass. It has to stand up maybe 12 to 15 feet tall, so it doesn't make wood, So it has to reinforce itself with these sclerenchyma cells so it can stand up that high off the ground and not blow over, okay? Parenchyma, thinnest, sclerenchyma, thickest of the cells. And then we can have complex or multiple cell types in tissues. Okay, those were simple. These are complex. So xylem, that part of the center of the stems that conducts water, these are made up of vessels and tracheids and fibers, and some of these fibers are sclerenchyma cells, and some of the tissue is parenchyma cells. So it's mixed, right? It's complex, multiple cell types in these tissues. Or phloem. Phloem is the other type of conducting tissue in plants that transports photosynthate manufactured foods from the leaf, and it takes it in various places from the roots to other leaves to developing fruits or seeds. It transports manufactured food and metabolites around the plant. So xylem transports water and minerals from the soil, and it's a one-directional transport. It's up. Phloem transports food that the plant makes in the leaves through photosynthesis and runs it all over the plant to feed different parts of the plant and metabolites and hormones and whatever else needs to go. So it's multi-directional. It goes up and down and sideways. And then together, of course, as I've mentioned, the phloem and the xylem make up the vascular system of plants, or you can think of it as the plumbing system, if you will, of plants. And if we look at a photograph here, this is an actual photograph of a cross-section of a plant stem. We see that there is xylem, there's multiple types of cells in that xylem band there, and same with the phloem. And you can see that in the xylem, some of those cells have thick red reinforced cell walls. They have to have that to withstand the pressures of pulling water up to such heights in trees. And then the phloem is the same. It carries different things, but the concept is the same. Okay. Now, if we look at a cube or a section of a leaf, you can see in the upper left-hand corner there's a leaf with a chunk cut out of it, and then we've stripped that away to show you the parts of the plant. You can see there's a big pink vein that runs down the middle of the leaf with little side veins coming off of it, and the veins are made up of xylem and phloem, and that's where they reside. Xylem and phloem don't live in the rest of the plant. They just live in the veins. And then you see on the far right, you see that there's upper epidermis, and then down below you see lower epidermis in the blue box there. Those are made up of parenchyma cells. The cuticle you see there on the upper left middle is a waxy deposit that's put down on every leaf surface to help with water loss. Otherwise, the plant would just lose water and wilt. If we go back to the right-hand side, we see palisade parenchyma and spongy parenchyma. Palisade means columnar, up and down, and spongy just means spongy. And there's two kinds of parenchyma cells there. Remember the thin-walled cells. They don't have to have a lot of mechanical reinforcement because they're inside the leaf. And then we see the pink are the collenchyma cells that make up the outside of the veins, and we talked about them in a mechanical function in the plants. If we look at a cross-section of a leaf, we see the big vein. This is flipped upside down. We see there's a big vein right in the middle of the leaf. You know, like a big corn leaf, let's say. There's a big vein that runs down the back side of the leaf. There are stomates and guard cells and cuticles there. We already know what the cuticle does. It's a waxy deposit that stops the plant from losing water. The stomate is an opening in the leaf, and the guard cells are cells, there's two, that surround the opening to the stomate that open and close and let water in and out and let carbon dioxide in and out. And it's for gas exchange and water vapor exchange. So if we look at a corn plant and there's some nice cocklebur, you notice the cocklebur is not showing any signs of being too dry, but you see that the corn plant, when it gets too dry, those leaves roll. And this is a characteristic of some grasses. A lot of grasses do this. Say, why do those leaves roll? What's the purpose of those leaves rolling? Well, does it increase photosynthesis? No, it actually decreases photosynthesis. They're not making as much food. So they roll because the stomates are located on the upper surface of corn leaves, and when they're under water stress, they roll to try to keep water, to raise the humidity inside those rolls, to shade it, to keep the hot wind from blowing on it, and to preserve water until such time water can be added to the soil either by rain or irrigation, and they will unroll and continue to photosynthesize and go about their business. So that's why they roll, is it's a response to drought. And the way it works is there are some cells inside this leaf. You see the leaf, that the upper surface is up and the lower surface is down with a big mid-vein. And then you see there's some cells in that highlighted circle there that are kind of large. Those are bulliform cells, and the bulliform cells hold the leaf out. It's when they're full of water, the leaf lays kind of outward and flat, and you'd call it normal. But when it's droughty, those bulliform cells lose their turgor pressure. That's their internal pressure, like the pressure in an air balloon or a water balloon even. When they lose that pressure, that allows the leaf to kind of roll in on itself. Okay, so it's like a party favor. When you blow it, it rolls, it unrolls outward. And when you stop blowing and the turgor pressure goes out of that little party favor, it rolls back up on itself. So when plants don't get enough water, they lose pressure and those leaves roll up on themselves. Well, there are so many kinds of leaves and stems. The picture on the left is a picture of a bamboo, a native bamboo grass that grows around here, reed if you will. And those are leaves that have rolled up there, and then some green stems. And whenever you see green on a plant, it's photosynthetic. It will be photosynthesizing. And then on the right is a picture, obviously, of a clover. And we've talked about clovers a little bit. Let's talk about the variation. And you think about cacti, right? And where are those leaves? Because you're mainly looking at stems. So what happens to the leaves of cacti, right? But those generally aren't crops, and so we're not really going to talk about those too much in this class. But there are so many kinds of plants and leaves and structures. It's what makes plants so fascinating to study. But let's talk about leaves. Let's talk about the leaf structure. Most leaves have a blade, which is the part that looks like the leaf. Then they've got the little stem that holds it up off the, or the little stalk that holds it up off the stem. That's called the petiole. And then sometimes at the base of them, they have a little bit of, looks like a little leaf or something there. That's called the stipule. Okay, the stipule. So we've got leaf blades, the petioles, and sometimes we have stipules. So there's the blade. And in this case, this is a clover. So actually this blade has been broken up into three parts: a center blade and a right blade and a left blade, but it's all considered to be one plant. The stipule is always at the base of the petiole, and the petiole defines the leaf. And so when you see that petiole, you know that you've seen the entire leaf. So there's the blade. This is the petiole. Okay, the petiole holds the leaf off the stem, and then here's the stipule. It's a modified leaf. It can photosynthesize. Not all plants have it, but when it's present, it does tell you that that's the base of the leaf. Because otherwise you might mistake this for having three separate leaves, right? But in fact, this is one compound leaf. Now, sometimes we can call that the petiolule, which is the little, it's like a little petiole, but it holds up the compound parts of the plants, of the leaves rather, the leaflets. So a petiole, let me make sure you understand this. A petiole supports a leaf. A petiolule supports a leaflet. Okay, this leaf has three leaflets or components to it. Okay, that should be clear to you. We can have, by the way, leaf morphology. Morphology means the shape of or the outline of. We can have simple leaves or compound leaves in our plants. This is an amaranth leaf. This is one of the pigweeds. This is a simple leaf. It's not broken up into separate leaflets or sections. It's just one big leaf, and that red at the far right is the beginning of the petiole that holds it up, that will attach it to the stem of the plant. Okay, so that's a simple leaf. Then I wanted to go back up to this picture here, and this shows you palmate versus pinnate. So this is a palmate leaf. It has 3 lobes, but they all meet at the base here, like the base of your palm. This leaf here on the far left is pinnate, right? So it's divided into little leaflets, and it looks like the pinnae of a bird feather. And then the one in the lower right is the same. Okay, so now we'll work our way back forward here. And then we're going to finish by talking about the unique anatomy of grasses and grass leaves. Not much. I'm not going to ask you to know all of this, but I am going to ask you to know a few pieces of this. So if you look at a grass leaf, and this is obviously a drawing of a grass leaf, you'll see that there's a stem that comes up through the grass, to which a blade, this blade right here, the blade is attached. But blades don't, in grasses, blades don't attach by a petiole. They wrap the stem down below in a sheath. And so here's the sheath right here, wrapping around the plant stem. And then we call this area where it transitions from the sheath to the blade, this is called the collar region. And that's all I'm going to ask you to know. That you don't need to know the ligule or the auricle. Just this is the collar region. So you have to know 3 areas: the blade, the collar region, and then the sheath that wraps around the stem. Okay. If you do get into grasses and you take some more advanced plant classes, the shape and arrangement and size and the dissection of this little ligule here and also the auricle will go a long ways to helping you identify which grass it is that you're looking at. But we won't do that in this freshman plant science class, but I would like you to know the blade, the collar region, and the sheath. So here's an actual picture of a corn plant. So here's the stem, the stalk we would call it here. Here's the blade, and the blade sticks out on the left-hand side of the picture. And then instead of attaching by a petiole, that leaf wraps around the stalk down below where the leaf exerts away from the stem. And you can see there's some hairs and things here. This is the collar region right here. And if we examine that collar region further, we would find identifying characteristics. But again, you don't need to know that for this particular class. Well, that's the end of the exam material for exam 1 in this class. It covers chapters or lessons 1, 2, and 3. There is a YouTube channel for these videos, and I've listed it right there. Those videos will be up on that YouTube channel now, or if they aren't already. Also, though, know that the PowerPoints are on Blackboard, and you can look at the PowerPoints and study off those PowerPoints. And then, when available, I always do the Cloud Zoom recordings for everyone. And they're also found on Blackboard. I do them for most classes, not all. Sometimes I can't. But if they're there, they're going to be found on Blackboard. So between the PowerPoints on Blackboard, the Cloud Zoom recordings, you attending class, and the YouTube channel videos, you should be in really good shape for the next exam. And this concludes this lecture.